I'm Jay Bradner, president of NIBR, which is the research and early development engine for Novartis. And I hope that all have enjoyed Breakthrough Science Media Day, a remarkable event where science meets hope, which is an apt description of our ambitions and especially this domain. It's a real pleasure to contribute to closing discussion. We've had already today an amazing consideration of the mid-stage portfolio, and wonderful to hear from you, Dr. Sai, as usual. Here, we'll meet with three leaders of our cell and gene therapy efforts at NIBR and take a deeper dive into these new modalities, sort of like a sneak peek into the pipeline and how we're thinking about these emerging therapeutic modalities, ever more a reality in the practice of medicine, to reimagine some aspects of these technologies. And the goal is that perhaps even through this reconsideration of the science of these therapeutics, that we can even democratize cell and gene therapy. These are medicines that can have a dramatic impact on patients, but how they can reach an ever expanding group of patients remains a real challenge for the field writ large, but one that we're working hard on. And so I'm really pleased to introduce and to have three of our top scientists here with me today, each of whom are leading efforts to overcome exactly the challenges that I've just outlined. I'm going to introduce each of the three of them now, and then we'll get to a discussion. First, Jennifer Brogdon. Jen is Executive Director and Head of Cell Therapy Research in the Department of Exploratory Immuno-Oncology at NIBR. Jen leads the development of next-generation CAR T-cell therapies for oncology, is responsible for building a large therapeutic pipeline from target discovery into first-in-human clinical trials. Jen helped drive the development of Kymriah, the first CAR T-cell therapy approved by the FDA, and has overseen 15 or more therapeutic immuno-oncology programs that have reached phase 1 and 2 clinical trials. Cindy Grosskreutz, a physician scientist who serves as global head of ophthalmology research at Novartis, probably someone you've read about or even written about in the last year. Cindy's been driving our work in optogenetics of late, including key developments to design therapies that create a new circuitry in the retina to transmit light signals to the brain where photoreceptor cells have been damaged by genetic or even more common diseases. These re-engineered retinal cells could potentially therefore replace photoreceptor cells that have been lost, and you'll hear more about this and other exciting work from Cindy's ophthalmology DA or disease area. And Sue Stevenson. Sue's an executive director in the exploratory disease area of our chemical biology and therapeutics department. This is sort of a technology skunkworks department where we take on some of the grand challenges in the science of therapeutics. And I'm sure you may recognize Sue's name if you've been following the news. Sue's been leading our new collaboration with the Gates Foundation to develop a one-time curative in vivo gene therapy for sickle cell disease. Now, we haven't made it yet, but we have a lot of pretty exciting ideas. But if we could do this, we could bring the promise of gene to cell therapy to patients in the most challenging parts of the world, those under-resourced countries where the need for cures for sickle cell disease is so pressing and immediate. All right, now I'm going to turn to our exciting panelists and scientific leaders, Jen, Cindy, and Sue. So I'm curious to know from each of you, how can we imagine, how will we invent a next generation of cell and gene therapies that will reach many more patients than we can possibly reach with current approaches today? Jen, first to you.
Thanks, Jay. It's a really great question. You know, with CAR T, it has really been a revolution for the cancer patients that have been needing something that's been truly definitive and transformative therapy. And I think what's interesting about it, it's a living drug that enables patients to fight their cancer using their very own immune cells. We're excited about the innovations that we've made in this space. I think we're really trying to leverage the biology of the therapy to its most fruitful benefit. And by minimizing the amount of work that we do with these cells, we're really enhancing their fitness and preventing them from getting exhausted. So when they go back into the patient, they have even more potential for proliferation, for deeper and more durable outcomes, hopefully overall better benefit and higher response rates. And as we've designed this new platform, because we've capitalized on the better biology of the product, we're giving much lower doses because this is a more potent therapy now. And we're hoping that by doing that, we can also avert some of the adverse events that we see with the first generation approaches. So as we continue to look at this, because we're shaving off time in how we manufacture these cells, we're looking to really accelerate that vein to door time, the time from which we collect the cells from the patient, manufacture, and get them back to the hospital, and looking to aim for less than half the time that it takes for conventional therapies now. So we really think this has tremendous hope for scaling up and out for all those patients in need.
Jen, thanks. The work to imagine a potentially curative cell therapy for blood cancers patients really resonates with me as a hematologist, but there are so many patients with solid tumors who are still wondering and waiting.
It is a long-standing challenge, and actually the trials initiated in solid tumors with CAR T therapy before we saw the success of the hematological cancers. We've learned a lot in past clinical trials, in glioblastoma as one of the indications where we started. I think this is a brain cancer, and what we learned is cells can get into the brain, but then they encounter a very hostile tumor microenvironment that prevents them from being able to expand and mediate their normal function. And so unlike CD19 target for B-cell malignancies, the targets for solid tumors often come with liabilities. They're frequently expressed on normal tissues, and this can pose significant toxicity risks. So one of the approaches we're using and we're exploring now is TCRs. And like CAR Ts, TCRs are still based on giving a patient's immune cells that have been modified to attack their cancer cells. But unlike CAR T, the TCRs can really zero in on proteins that are inside the cells. And you can think of mutant cancer proteins that are driving the disease that can be targeted in this fashion. So there's a number of reasons we think that this might be better suited to solid tumors. And I think one of those primary reasons is now we can go after tumor-specific antigens to really minimize the risk of normal tissue toxicity. And hopefully, the ultimate goal is to find new antigens that are shared across different tumor types that could lead to broader therapies that might work across multiple cancers.
Thanks, Jen. Cindy, let me ask you some questions about this idea of expanding the impact of gene therapy in ophthalmologic diseases. These gene therapies have sort of grown up in the inherited retinal disease space where, estimates of 1 in 2,000 individuals may suffer from one of these diseases, but there's like 250 or more of them. Mm-hmm. And then there's so many patients, the vast majority of patients who suffer vision loss that don't have a single monogenic cause for their vision. How can we bring gene therapies to this massive and aging and prevalent population of blind patients? What do we need to do to reach more patients with blindness?
So I think that you touch on some of the excitement and the challenges in the field. The excitement for gene therapy in ophthalmology is that it's been shown for the first approved therapy that replacing a gene in a patient with a genetic mutation in that disease can actually benefit their retina. But as you say, there's over 250 of these different genetic mutations, and it's impossible to believe that we would develop 250 different types of gene therapies. And then, as you say, they're all the patients who we don't know what causes their retinal degeneration. The one thing they all have in common, though, is that the photoreceptors, or the cells in the retina that detect light, die. And the approach that we are now taking is to try to bypass those cells altogether. Those cells are going to die. And if we can't save those cells, what's next? What's next is trying to take other cells in the eye that are surviving. Not all cells die when the photoreceptors die, and use gene therapy to introduce a genetically engineered light sensitive protein to those cells so that they can take over the light detecting function that has been lost with the photoreceptors. This has the potential to be completely gene agnostic, so it doesn't matter what the mutation is, and to extend to the patients who we don't know what their genetic mutation is. It's incredibly exciting, and I think will expand the number of patients who have a hope of having a therapy dramatically. There are a couple of other things, though, that are, I think, important about this approach. One is that the field of optogenetics continues to evolve and improve. Initial work in the optogenetics field were using some genetically engineered proteins that were sensitive only to very, very bright light. And in those approaches, patients needed perhaps light boosting goggles in order to help them see. The approach that we're taking right now utilizes a genetically engineered light sensitive protein that is much more sensitive to light. And the goal of that is to allow a patient to potentially see a loved one standing right in front of them without having any type of light boosting goggles that would go along with it.
You know, I love this as a committed drug hunter, and just the idea of, reimagining what it means to be a therapeutic. I mean, to fully re-engineer tissues for potentially definitive therapeutic benefit. I also really admire the openness to engage with experts in a really rapidly moving field, to complement external technologies with hard-won internal insights and platforms, and moreover, the truth-seeking mindset of NIBR gene therapy research. Thank you. Sue, your pioneering work on blood stem cell biology also invokes an engineered solution. How does this work? And when it works, how will we reach patients around the world with sickle cell and thalassemia, where so many live in under-resourced healthcare environments?
Right, great. Thanks, Jay. So sickle cell is a complex genetic disorder that affects the structure and function of red blood cells. One of the treatment options for sickle cell disease is actually a stem cell transplant. So what the field has been pursuing is an ex vivo modified autologous cell therapy approach to cure sickle cell, either with a gene replacement or with CRISPR editing. We at Novartis are actually in the clinical studies right now with CRISPR genome edited stem cells as a therapeutic approach for sickle cell. And this involves taking the hematopoietic stem cells outside of the patient's body, modifying them in a lab, and then after modification, transplanting those modified cells back into the patients. Now, as you heard from Jen, any kind of ex vivo autologous cell therapy is complex. It requires a specialized infrastructure. And because of that, it's not going to be accessible to a large percentage of patients because of the complexity involved, one, but also it requires a stem cell transplant. And the way that you have to then deliver those modified cells back into the patient requires a stem cell transplant with toxic conditioning regimens to allow those modified stem cells to engraft. And so the field and these ex vivo cell therapies are showing real promise. And so we're trying to build on the foundation that we've established with this ex vivo therapeutic approach for sickle cell, with the mechanisms and understanding what it takes to develop a curative therapy, and to think about envisioning an in vivo therapy. Rather than take the hematopoietic stem cells outside the body and modify them and transplant them back, we want to target those hematopoietic stem cells inside the body. And that requires sophisticated delivery, innovating the delivery of how we're going to modify those hematopoietic stem cells. Now, an in vivo therapy, again, would be a one-time administration without the need to modify cells in a lab or require long hospitalization stays, as is required now for a stem cell transplant. And so because of that, that would enable many more patients to be treated with this therapy, given the safe and selective delivery to hematopoietic stem cells inside the body. But most importantly, it'll allow us to think about treating patients where they live. And as you know, Jay, 80% of all patients, sickle cell patients, live in sub-Saharan Africa. So how do we envision taking a therapy, an in vivo gene therapy, to those resource-restricted countries? So very excitingly, we've recently have established a collaboration with the Bill and Melinda Gates Foundation to do just that. So this collaboration brings the best of the Gates Foundation with the best technologies and drug hunters within Novartis to bear on this aspirational goal of in vivo therapies that can be more globally accessible. We have a dedicated team focused on this that would be first innovating the delivery of how we're going to target and gene edit those hematopoietic stem cells inside the body first. Second, we're going to innovate manufacturing. So we're going to eliminate the need to take the stem cells out by targeting the stem cells in the body. But that's going to require a more innovative manufacturing protocol, if you will, for that targeted gene therapy. And then we're going to start doing clinical studies in these, establishing the infrastructure and interaction with health authorities in these certain countries that we can target where the majority of patients live. And then we're going to plan for early access. And again, using the networks and infrastructures that the Gates have established, we'll be able to enter these countries and to bring these definitive, potentially curative therapies to where the majority of patients live. This is an exciting collaboration. I'm excited to be leading this collaboration and with this aspirational goal. It's really early days. We've just started, and we're focused right now on trying to figure out how to target these stem cells and what vector systems should we innovate. And we're taking advantage of all of the technologies and innovations that we've established here within Novartis and leveraging those manufacturing platforms in order to develop these in vivo gene therapies. So really exciting time, and we hope to be progressing forward in the next several years with our first targeted in vivo gene therapy.
Well, thank you, honestly, all three of you for sharing this important and exciting science. I have just one last question, like a speed round for the three of you. I mean, having all three of you here together, it's such a pleasure. And I know your leadership teams work really closely together through our global cell and gene therapy initiative. These fields are moving so fast. You know, new CRISPR enzymes are published all the time, new capsids with different tropism reported each week in the scientific literature, in fields that are moving so fast. How do you keep up? How do you leverage learnings from each other's groups? And concretely, what learnings in cell therapies have contributed to our viral gene therapy work and vice versa?
I'm going to answer that in two different ways. One is a more general way that I think we've learned as a group in cell and gene therapies. And it is that many of these things were innovated in very high quality academic laboratories. And there is a step between that and giving it to a patient, which is how can we safely manufacture? How can we get these agents to live up to the promise that they had in a laboratory when we put them into patients who need them? Safety has to come first and has been really the driving principle behind a lot of the manufacturing and development. In terms of getting it to a broader swath of the patient population, educating physicians, educating patients, working with patient advocacy groups, and understanding that gene therapy is not a pill that you just take every day. It is a one-time treatment by and large, but that there are many subtleties and nuances that are inherent in delivering a gene to the body of a patient that are very different from the small molecules that we all take every day when we take a pill.
It's fair to say that at NIBR, we love cell and gene therapy research. And we remain committed to this challenging, uncertain, expensive research because these medicines can be curative. They so matter for the patients that they have to date successfully reached. We start these projects knowing just who the patient is at the very earliest moments of the science. We love them because each is an ensemble of dozens, if not hundreds of innovations built on a really strong basic science foundation. They're so modular. They're ideally suited to science at the intersection of domains of deep expertise as we enjoy at NIBR. and in our surrounding scientific communities. And make no mistake about it, we cherish this chance to reimagine medicine as re-engineered cells or gene-bearing viruses because it's hard. It's well suited to a research environment like ours that really strives for this highest hanging fruit. Gene therapy and cell therapy are truly where science meets hope, and so apt then to consider in today's conversation. Thank you for your interest. We look forward to sharing insights and advances from the cell and gene therapy platforms and the rising clinical programs in the research portfolio, all in the months and years to come. Take care.